Clinical Chemistry Introduction to Enzymes. Enzymes are specific biological proteins that catalyze biochemical reactions without altering the equilibrium point of the reaction or being consumed or changed in composition. So they're biological proteins. They catalyze, which means they help change or modify biochemical reactions, but they don't mess with the equilibrium point of the reaction, and they're not going to be consumed or changed during this process. They're found in all body tissues, and they frequently appear in serum following cellular injury or degradation. So a lot of enzymes, especially enzymes that are specific for certain organs, those are going to be found within the cell. If that cell is damaged, if it ruptures, those enzymes are going to be released, and then we see a peak in those enzymes in the serum. So if they're supposed to be inside of the cell and the cell breaks open, then we can measure them in the serum, and that can give us an indication of which organ or tissue has been damaged. An enzyme may exist in different forms too. An isozyme and an isoform are different forms of enzymes. Enzyme structure, so there's an active site to the enzyme, and the active site is the physical location where the substrate binds for catalysis. So the substrate is going to interact with charged amino acids of the active site, and it's going to be able to bind. We used to refer to this as a lock and key fit for the substrate to its active site. Now we think of more of an induced fit model. When the substrate comes close to the active site will modify its structure just slightly so that the substrate can fit into the active site. Allosteric site is a cavity that's other than the active site where the regulatory molecules bind, and this affects enzyme structure. So if a regulatory molecule is going to bind to an allosteric site, the enzyme will change its shape. Sometimes that means that that enzyme will no longer be able to interact with certain substrates. A proenzyme is a structurally inactive form of an enzyme. Other enzymes that alter the structure of a proenzyme to make it active sites available for catalysis. Also termed zymogens, that's common amongst digestive enzymes. So in the digestive system, we typically are going to release inactive enzymes, and then there's substances in the digestive tract that will activate them to form an active form. An example would be a pepsinogen and pepsin and hydrochloric acid. So pepsinogen is released, and then pepsinogen is converted to pepsin by hydrochloric acid. And then pepsin is going to be the active form of the enzyme, and that's going to break apart proteins. So that's just one example of how zymogens or proenzymes are going to work. There's also cofactors that are going to affect enzyme function. So it's a non-protein molecule that's necessary for protein activity. An activator is an inorganic cofactor, such as calcium or magnesium. There's other types of metals that can serve as activators. A coenzyme is an organic cofactor, so it's a little bit larger than, much larger than an activator, and it's also going to have carbon in it. So NAD and FAD are examples of coenzymes. A prosthetic group is a cofactor that's tightly bound to protein. An apoenzyme is the protein portion without the cofactor, and the holoenzyme is the whole enzyme. So it's the apoenzyme plus the cofactor, and that's the complete and active enzyme system. Catalytic mechanisms of enzymes. So the activation energy is the energy required to raise all molecules in 1 mole of a compound at a certain temperature to the transition state at peak of the energy barrier. So the activation energy is the energy that's required in order to make a reaction go. Enzymes catalyze reactions by lowering the activation energy level. So they lower the amount of energy that's required in order for this reaction to proceed. Enzymes don't necessarily do something that wouldn't already happen, but they increase the rate at which it can happen by decreasing that activation energy. Enzyme specificity can be different. Absolute specificity means that it's specific to only one substrate. So one enzyme can only act on one substrate. Group specificity means that it's specific to all substrates of a specific chemical group. Bond is specific to a type of chemical bond, and stereoisometric is specific to one optical isomer of a compound. Energy versus progression of the reaction, indicating energy barrier that the substrate must surpass to react with and without enzyme catalyst or catalysis. So we have here is without an enzyme, it requires this much energy in order for it to proceed with the reaction. With the enzyme, it's going to require this much energy. So it decreases the activation energy so that the reaction can proceed. And the reaction that we're talking about here is enzyme plus substrate to produce product. The Michaelis-Menten curve defines the relationship between the substrate and the velocity of an enzyme reaction. So we can see that substrate concentration is over here and velocity is up here. So as we increase substrate concentration, we're going to increase the velocity of the reaction up until a certain point. And after that certain point, we're going to start running out of enzyme. So no matter how much more substrate we add, we're not going to increase the speed of the reaction because we have run out of enzyme at that point. Factors that influence enzymatic reactions. The substrate concentration. So if we only have a little bit of substrate, we're not going to have very good enzymatic reactions. Enzyme concentration. Again, if we only have a little bit of enzyme, we're not going to be able to have enzymatic reactions. The pH. So the pH affects the protein folding. So proteins are going to have different properties when they're in different pH levels. So proteins are going to take on different charges depending on the pH that the protein's in. So when proteins interact with the pH of the environment, they are going to change their shape. So they can only be functional within a certain pH range. If we change the pH, that enzyme will no longer be functional because it's going to be unfolded because the charge of those amino acids is going to be altered. Temperature is another thing. So temperature, if you increase temperature, you're going to increase the rate of the reaction. However, beyond a certain temperature, the proteins are going to denature, so they're going to unfold and break apart. So temperature plays a major role in the activation of enzymatic reactions. The availability of cofactors is another one. And inhibitors. Inhibitors are going to prevent an enzymatic reaction from happening. There are competitive, non-competitive, and uncompetitive inhibitors. Competitive inhibitors physically bind to the active site and compete with the substrate. They're structural analog of the substrate. So they are going to fight the substrate to try to get into that active site. Non-competitive inhibitors are going to bind to the enzyme at a site other than the active site. It may be reversible or irreversible binding, but if that inhibitor binds to the allosteric site or the site other than the active site, it is going to change the shape of the enzyme, and then that enzyme will no longer be able to be functional. Uncompetitive inhibitors bind to the enzyme substrate complex. Increasing substrate results in more enzyme substrate complexes, and that binds more inhibitor, which increases the inhibition. So how do we measure enzyme activity? Common measures include an increase in product concentration. So we're going to measure the amount of product that's produced, and then we can say that if it increases, we have increased enzyme activity. A decrease in substrate concentration. So if we measure the amount of substrate that's present and we have less substrate than we did before, then we can say that the enzyme is active. A decrease in coenzyme concentration. So if the coenzyme is being consumed, then we know that the enzyme is active. Or an increase in concentration of an altered coenzyme. Enzyme concentrations are always performed in zero-order kinetics. Inhibitors must be lacking and all other variables carefully controlled. The types of measurement of enzymatic reactions include fixed time, continuous monitoring, and kinetic assays. Calculation of enzyme activity. Enzyme concentration is usually expressed in units per liter. The international unit is the amount of enzyme that will catalyze the reaction of 1 micromole of substrate per minute under specific conditions or under the specified conditions. Measurement of enzyme mass is done by immunoassay methodologies. There is a risk of overestimating the active enzyme due to possible cross-reactivity with inactive enzymes, though. Enzymes as reagents, so enzymes used to measure many non-enzymatic constituents in serum. There is a naming system to the different types of classes of enzymes. So the IUB system is a numerical code of 4 digits that's separated by decimal points. There are 6 different classes of enzymes that are recognized: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. You need to be familiar with this entire table, their number, class, and reaction that is catalyzed. Several questions will come from this table on the test. So just an example of the classification and nomenclature. Lactate dehydrogenase, the common abbreviation is LDH, the standard abbreviation is LD, and then it's given an EC code. So the EC code of 1 means that it's in class 1. And then 1.1.1.27 is the full code. And then it's given a systematic name, L-lactate NAD oxidoreductase. Aspartate aminotransferase, the common abbreviation is GOT, glutamate oxaloacetate transaminase. The standard abbreviation is AST, and the EC code is 2.6.1.1. So if we go back to 2, it's a transferase, transfer of group other than hydrogen. Systematic name is L-aspartate 2-oxoglutarate aminotransferase. You do not need to know the details of these names. These are just examples so that you can understand the different ways that we would name these enzymes.